Good day, everybody. Today we are here to introduce to you our work on mitosis, as well as for you to learn more information about the subject assigned to our group. Components that are included in the flow of presentation composes of the introduction, the members' designation of roles of the project, making of the project, the development of the project, as well as the coordination of each participant in the project's construction. And it was all made possible by Team G's passion, knowledge, and teamwork.
The task comprises of answering the challenging question, the model-making process and its stages, as well as a brief reflection on the activity. The PBL began with a challenging question that each team had to answer. Sometimes you accidentally bite your lip or get wounded, but in a matter of days, the wound heals. How does it happen? Why do you think there is genetic diversity that we look different from each other? How will you explain the process?
Hello everyone, we're Team G. I'm Andrea Nicole.
I'm Cristha and Santos.
I'm Amanda.
Our group was tasked to create a 3D model about mitosis.
Today we are here to introduce to you our work on mitosis, as well as for you to learn more about the information and the subject assigned to our group.
Have you ever wondered if you could clone yourself? Well, you can't do that. Actually, there are some really good reasons why you can't. But do you know what can clone themselves? Your cells. Like almost every single one of them. And in fact, they're doing it right now.
For any creature bigger than a single-celled organism, all of life stems from cells' ability is to reproduce themselves, because that's what allows organisms to develop, grow, heal, and keep from dying for as long as possible.
This particular kind of cell division is called mitosis, and it's responsible for a whole lot of our body's key function. By now, you can see that mitosis is essential in replacing worn-out cells and healing tissue damage. When you grow from a 7-pound baby to a 70-pound child, it's not your cells that are increasing in mass, you're just getting more of them.
Mitosis is so central to your life that it will take place in your body over your lifetime about 10 quadrillion times. That's 10,000 billion times.
So you are made of trillions of cells just like giraffes and trees. And remember that inside each cell there's a nucleus that stores your DNA, which contains all instructions on how to build you.
That DNA is organized into chromosomes. In your body cells or somatic cells, you have 46 chromosomes grouped into 23 pairs. One in each pair is from your mom, and the other one's from your dad. This makes sense when you realize sex cells, the egg and the sperm cells, only have 23 chromosomes each.
These are called haploid cells because they have half as many chromosomes. But cells with all 46 chromosomes, somatic cells, are called diploid cells. And that's what we are focusing on today. Now, the nucleus in your cell controls everything that goes on in the cell. It has all of the instructions necessary for making the cell survive, so you don't need to duplicate the whole cell. All you need to do is duplicate the DNA, get it wrapped up, and then if you have two separate packets of DNA, that's all you need to have two new cells.
The main thing to remember about mitosis is that it allows one cell with 46 chromosomes to split into two cells that are genetically identical while still having 46 chromosomes, all in order to keep the party of life going.
Mitosis takes place in a series of discrete stages called prophase, metaphase, anaphase, and telophase. But let's begin with what we call the interphase. Interphase is the part of the cell cycle where the cell is not dividing. Think of this as normal cell life. The cell is doing normal cell things, growth, metabolism, and so on.
During interphase, the long strings of DNA are loosely coiled and messy. This stringy, uncoiled form of DNA is known as chromatin.
One of the more important things that happens is that the little set of protein cylinders next to the nucleus called the centrosome duplicates itself. They're gonna be the ones with the spindle fibers later on.
But the really important thing that the cell does during interphase that is needed in order to do mitosis is that all of the DNA begins to replicate itself too. Notice that each piece of chromatin has now a copy.
One final thing I want to make clear is that interphase is not part of mitosis. It happens before mitosis and is especially important because it is where the cell's DNA is copied.
Now the cell enters the first phase, or the prophase, when that mess of chromatin condenses and coils upon itself to produce thick strands of DNA. Then the nuclear envelope gets out of the way by completely disintegrating, and spindle fibers form as microtubules grow out of centrosomes that move to opposite poles of the cell.
Now we have reached the metaphase, which literally means the after phase, and it's the longest phase of mitosis. It can take up to 20 minutes. During the metaphase, the chromosomes attach to those ropey microtubules right in the middle at their centromeres. Now, the microtubules are attached to each sister chromatid. The double-stranded chromosomes line up along the equator of the cell, and the spindle is now fully formed.
Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the X-shaped chromosomes split back into their individual single chromosomes.
Once they're detached from each other, they're dragged toward either end of the cell. The prefix anum means back. That may help you remember the name of this phase. That's again anaphase.
After this, it's just a matter of using all of that genetic material to rebuild, so that the copy genetic material has all of the accoutrements of home.
In the last phase, which is telophase, each of the new cell structures are reconstructed. First, the nuclear membrane reforms and nuclei form within them, and the chromosomes inside relax back into the chromatin. The spindle also disappears.
Then, a little crease forms between the two new cells, which marks the beginning of the final split. That division between the two new cells is called cleavage. All that's left is to make a clean break. This is done by cytokinesis, literally cell movement, by which the two new nuclei move apart from each other and the cells separate. We now have two new cells, each with a full set of 46 chromosomes.
In this project or activity, Team G built a model showing and describing stages of mitosis. The purpose of this activity is to explain how cells multiply and replace damaged cells. This activity also proved what we have learned during the last activity, that all cells come from pre-existing cells. Mitosis is a process where a single cell divides to form two identical cells. Because we were tasked to make a model, here are some of the things we used to achieve that. A laptop or computer for researching, editing, and communication, a band paper that we used as a base to where the model will be, a color pen for adding colors or interpretation, a rubber band used as a spindle post in the model. We also used folders that were used as the cytoplasm or plasma membrane in the model, blue and green clay used as the chromosomes or chromatids in the model, purple or pink yarn used as the mitotic spindle fibers in the model, masking tape to place everything neatly on the band paper. Through this activity, we were able to understand how mitosis works better because of the 3D model we made. This helped us to answer the challenging questions better and digest the point of the project.